going to talk about mechanisms of speciation and mechanisms of isolation that allow speciation to occur. So first of all, what is speciation? We've talked about it before, but just to refresh your memory, speciation is the process of species formation. We can talk about microevolutionary processes that lead to population divergence to produce new species. You should be familiar with the idea of microevolution leading to macroevolution and therefore speciation. But microevolution is the change in allele frequencies that occurs over time within a population. We've spent the past couple weeks talking about speciation, microevolution, and allele frequencies in terms of population genetics. And here we'll just wrap it up with a tight little bow and put all of that information together in a nice synthesis. So in terms of the processes behind microevolution, we have mutation, selection, both natural and artificial. We have gene flow or the lack thereof, and genetic drift. These terms should largely be review for you since we covered this in our population genetics module. So first things first, what is a species? We keep throwing this term around, species, population, but it might surprise you to learn that the argument over what a species is something that is alive and well in scientific communities. It's becoming more and more evident that what we tend to associate as species may not be species. They may be subspecies or populations. We are ever increasing our ability to obtain genetic sequences for things, and that has led to a revival in understanding what a species is. But for now, we're just going to go over the main species concepts that help scientists define what a species is. So first of all, we have what's called the morphological species concept. This is based on differences in anatomical features. You all have been exposed to the word morphology before, so therefore you should understand the morphological species concept is talking about anatomical physical differences in body plans. This is the only species definition that is available for fossils. So it might be easy for us modern day biologists to talk about genetic sequencing and high throughput sequencing analysis and next generation sequencing. You can't really do that for fossils. You only have their body plans and the shape and size and different morphometric analyses that you can do on a physical specimen. You can't obtain genetic material from a fossil unless it's very recent, and that just does not happen. So problems with the morphological species concept, there are several. Number one, some anatomical differences occur within the same species, and we're actually going to be discussing that today in terms of snail morphology and a specific case study that I found this morning. Number two, some separate species are identical in appearance, which makes this definition very, very hard to implement. And then finally, our morphology is not always evolutionary based. As we learned several lectures ago, morphology does not always equal homology. Just because you look the same in body plan doesn't mean that you are closely related. We can bring up again the example of the Tasmanian wolf, which is a marsupial, and how it looks the same in body plan and size and shape to the modern day wolf that we are more familiar with. That is a placental mammal. Instead of carrying the young in a pouch like marsupials, it has a placenta inside. Even though those two species look the same, they are not closely related. Okay, so again, morphology does not equal homology. The diagnostic characteristics for the morphological species concepts gets a little funky. So here we have two warblers. We have the yellow-throated warbler on the left and the myrtle warbler on the right. These are two distinct, clearly defined species. However, the only differentiating characteristics that we can see are based on patterns of color, both in the tail region here and in the throat region here, okay? So their distinguishing diagnostic characters or characteristics are based on feather color. Now, if you were trying to separate these two organisms based on the fossil record, you would never think that they're two distinct species because unfortunately pigments degrade very rapidly in the fossilization process. We are very happy to get imprints of feathers, but it is very, very, very rare that the pigments in those feathers remain preserved. Therefore, if we were to see these two different species preserved in the fossil record, we would never be able to identify them as two distinct species according to the morphological species concept. Moving on to the biological species concept, this is one that is more robust and accepted. This is taking the stance that if species interbreed in a natural setting, not a zoo, but in a natural setting, and they have viable offspring, they are regarded as the same species. Okay, so this is based on reproduction and the ability to have viable offspring, meaning your offspring can also in turn have offspring in the next generation. So thus, the biological species concept is based on reproductive isolation. If populations can interbreed, they are members of the same species according to the biological species concept. But if a population does not make fertile offspring, they are considered different species. Okay, there are, of course, problems with this. What about asexual organisms? We've learned about some of those at the beginning of this class. Also, what about extinct organisms? Again, reproductive isolation is not something that's necessarily preserved in the fossil record. Next, we have the phylogenetic species concept. This marries together both the morphological and genetic data that is driving the biological species concept. So we've used the term before, phylogenetics and phylogeny, to evoke the sense of a tree of life, something like this. But it may not surprise you to learn that genetic data is difficult, but again, not impossible to retrieve from fossils. And the more data you have, the more accurate the phylogeny that you can build is. And so the phylogenetic species concept has gained traction with the advent of next generation sequencing technologies. But again, it's kind of leaving behind those paleobiologists and paleoecologists who study organisms preserved in the fossil record. Let's talk about geographic isolation and how that plays a role in speciation. We've used this term before, subspecies, and this is simply defined as local variants of a species. Subspecies can technically interbreed, but they have different selective pressures. We've also been exposed to this term before, ring species. Ring species have genetic variation around uninhabitable terrain, and distant populations exchange genes through intermediaries. We've been exposed to the idea of ring species before in discussing different breeds of dogs. So the Great Dane and the Chihuahua, they are probably not going to be copulating together much, but if you input something that's a medium-sized dog, like a beagle, for example, that beagle can then reproduce with the Great Dane and the Chihuahua and have an intermediate stage in the beagle. So a ring species is genetic variation around uninhabitable terrain, or there's some sort of other reproductive barrier like size. It makes it hard to copulate if you're a Great Dane and a Chihuahua. And then finally, clines. This is genetic variation across a geographic gradient, and we'll talk about each of these in detail in the coming slides. Subspecies. This is an example I was only recently made aware of. We have Ursus americanus kermodii. This is a subspecies of the American black bear, but as you can see, it's all white. It's not a polar bear. It's a subspecies of the American black bear. We can find this particular subspecies in central and north coast regions of British Columbia in Canada. It's pretty rare. It's also called the spirit bear because they're so rare to find. There's high value placed on spotting them in the wild. Here's a different example of ring species. Again, these species or different populations are connected through a series of neighboring populations. They are able to interbreed through intermediates, and they have two end populations. And there's lots of gene flow between closely located populations. So first here, taking a look at a species of salamander in California. We can see that we have the mountain range here in the Great Basin and Range of California. Different colorations here represent different subspecies of the salamander species. So you can see it actually forms a physical ring around the tough geographic terrain of the mountain range here. So all of these populations have different colors, and some are a little bit chunkier than others, and they have different physical appearances. However, they're able to interbreed between the two end populations through intermediaries found in this ring. Another example here is the herring gull and the lesser black-backed gull. These are two phenotypes with clear differences in their physical appearance, but they're actually able to interbreed in a ring around the North Pole. And I think this is so fascinating. So here we're looking at the top of the world. We've got the North Pole right here at 90 degrees north. We have 1, 2, 3, 4, 5, 6, 7 different populations, and they're all able to interbreed with the neighboring populations in a physical ring, with the geographic barrier being the high cold temperatures in the high Arctic here. So very fascinating stuff. Next is something called clinal variation. This is when we have distinct populations along different geographical gradients. These geographical gradients can vary in their scope and size. The most common are latitudinal clines related to temperature and day length, where you get very, very long days or very, very short days, depending upon how far away from the equator you are. And of course, being warmer at the tropics than at the poles. We also have on a smaller scale, so not a global scale like latitude, but on smaller scales, this altitudinal cline. This is when we're dealing with mountain slope terrain, for example, and differences in humidity or partial pressure of oxygen, for example, as you go up and down a mountain slope. So here's an example with the sparrow in North America. We have different populations that are following this latitudinal cline. So lines of latitude are those that run horizontally. And as we can see, we have different shifts in the populations going from southern North America to North America in Canada. Something else that we should consider as a cline is maybe something that doesn't immediately pop to your mind, but differences in human impacts, for example. So this is a great example of the peppered moth, and we've talked about the peppered moth in previous lectures, but this is a different type of cline in industrialization. So the degree to which there was pollution in the air, that is in itself a cline or a gradient. And so pre-industrial revolution, we had two morphs of this peppered moth. We had the black and the white, with the black population being preyed upon more because it was really bad at camouflaging against a white backdrop of white bark. However, with the increase in soot and carbon emissions post-industrial revolution in the UK, we see that we increase increase the amount of the Black population while simultaneously decreasing the amount in the white population. And that's because we changed the environment. The environment shifted from being favorable to camouflaging those white individuals to being favorable in camouflaging Black individuals. So we see practically a one-to-one relationship in the increase in abundance in our Black populations compared to a decrease in abundance of the white population of the peppered moth here. And this is an example of human influence, and specifically talking about 19th century United Kingdom when we had the industrial sector boom. So clinovariation takes a wide array of forms, and this is one that I didn't really think about before. I only put it in terms of latitude and temperature and humidity and altitude, but really human impact is becoming a more and more influential cline. So let's talk about reproductive isolation. This comes up with the biological species concept and the phylogenetic species concept. So what do we mean by reproductive isolation? Reproductive isolating mechanisms prevent gene pools from mixing. Remember, the gene pool is just the aggregate of all alleles in a population. So this reproductive isolation prevents animals from breeding together. There are two different forms of reproductive isolating mechanisms. We have pre-zygotic barriers and we have post-zygotic barriers. And this is just what it sounds like. So pre-zygotic barriers occur before zygote formation. This is before fertilization can even happen between two individuals. By contrast, post-zygotic barriers occur after zygote formation, after fertilization has already taken place. So let's delve into specific examples of pre and post zygotic barriers. Pre-zygotic barriers include things like habitat isolation, temporal isolation, and behavioral isolation. So first, habitat isolation has to do with physical distance. So if you're in a population over here that's on one side of a cliff, and you have another population on the other side, there's a great expanse of distance between you. It makes sense that you're going to mate with things that are in your local area, organisms that are in your local area, compared to making the long journey of large physical distance away. So this is habitat isolation, where populations are found in different habitats, and it's much simpler to mate with organisms around you in your immediate surroundings. We have something called temporal isolation. This is when different mating schedules are being used. Here's an example between two related frog species that exhibit temporal reproductive isolation. The Rana anora in this A panel here, it breeds earlier in the year than Rana bollei. Okay, so because these organisms are breeding much earlier in the year than this organism, there's no chance that they're going to try to reproduce with each other. This is a pre-zygotic barrier. And finally, behavioral isolation due to differences in mating signals for sexual selection, for example. And a nice example of behavioral isolation are different types of fireflies that are attracted to specific patterns of flashes and pauses. And if you're not emitting the right pattern of flashes and pauses, you're not going to attract the mates. So there are different mating signals that are attracting different types of females or males, and therefore we have behavioral isolation. We also have mechanical isolation as a prezygotic barrier. This has to do with incompatible mating structures or reproductive organs. Here's an example figure in your textbook, figure 18.19. This is depicting the shape of male reproductive organs across damselfly species. They're only compatible with females of the same species, and reproductive organ incompatibility keeps the species reproductively isolated. So here we're talking about all of the same order, of insects. They're all damselflies. But because the male's penises are shaped differently, there's no compatible way to have a damselfly of this species copulate effectively with the damselfly of this species. Okay, so the female reproductive parts are directly compatible with the male reproductive parts within the same species. Okay, you can see that they're shaped vastly differently. They're sometimes longer or shorter. They have different appendages on them. And so therefore this is a mechanical isolation because the male reproductive parts are going to be incompatible with female reproductive parts of a different species. Moving on to post-zygotic barriers to isolation or reproduction. This is something that happens after copulation and zygote formation. Number one, we have gametic incompatibility or hybrid inviability. This is when hybrid individuals in many cases can't form normally in the womb and simply don't survive past the embryonic stages. So there was able to be a copulation event, a zygote formed, but it is not carried to term and therefore it dies in the womb. So this is gametic incompatibility. Most of the time this has to do with differing numbers of chromosomes in the genome, and therefore it doesn't become developed as a normal species would. Number two, hybrid sterility. This is when the offspring can't have viable offspring of their own. An example of this is the mule or the zonkey. Guys, you can breed a donkey and a zebra, and it is called a zonkey, okay? This is an actual thing. You can Google it, but it's amazing. However amazing it is, though, it is sterile. So this organism, this zonkey, cannot produce more zonkies, unfortunately. I would love to have a zonkey. I think my dogs would play with it so much. Anyway, okay. Oh, the zonkey. Okay. Number three is hybrid breakdown. This is when the hybrids between two species have reduced fitness. Hybrids will have fewer offspring and thus are likely to be outcompeted by those bona fide species in the surrounding area. So here we do have viable hybrids that are able to reproduce on their own. That's great, but they have reduced fitness in comparison to the bona fide species around them, and therefore they can't compete really with those species. Here's a table for you that discusses the timing relative to fertilization, whether it's a pre-zygotic or a post-zygotic mechanism. Here we have the specific mechanisms that we just talked about, and then the mode of action that we spoke about. Okay. So next, getting into the geography of speciation. And this asks the question, in what ways does speciation occur in terms of animal distribution on the planet? So that's a big question. We're going to talk about three different types of speciation. We have allopatric speciation, sympatric speciation, and finally parapatric speciation. So first up, allopatric speciation. Allopatric speciation results from a group of organisms being separated by physical geographic barriers. As I love to do with big biology words, we can break this down. So allo, the prefix here means other, and patric means homeland. Okay, so allopatric speciation is other homeland speciation. Here we have an example of the northern spotted owl and the Mexican spotted owl. And they are two different subspecies that had a common ancestor in recent evolutionary history. However, because of the mountain building event that created the basin and range structure here in the western United States and the Rocky Mountains, caused physical separation between the two populations, and therefore they're now their own subspecies. So in the example of this owl, there was a physical barrier the mountain range that prevented gene flow from occurring between these two populations. We had the buildup of subsequent mutations and adaptations that change each group. So we have two different subspecies of our owls here, and it's interesting because the northern spotted owl has genetic and phenotypic differences from its close relative in the south. So we can take a look at both their physical appearance, and we do see that these two subspecies of owl look physically different from each other, but we can also see those differences in their genomes. And it's evolution that's happened spectacularly fast because this is a relatively recent mountain. Okay, I just looked it up because I was curious. The mountain building event that produced the Rocky Mountains happened 55 to 80 million years ago. So this is pretty fast in terms of evolution. The allopatric speciation process involves two processes, dispersal and vicariance. Dispersal is when a few members of a species move to a new geographic area. And vicariance, by contrast, is when a natural situation arises to physically divide organisms. So looking at the two examples here, we both have in these examples geographic isolation, but dispersal is when a few members decide that they're going to go colonize a new geographic area. So here we have a figure that is depicting these two scenarios that are promoting allopatric speciation. On the bottom, this is representing dispersal. So we have an ancestral population of a species. We call it species A. And then over time, some representatives from species A decide to colonize a small island that's across the river. We can call this population species A prime because it's still able to interbreed with species A, but it's on its way to becoming geographically isolated. Over time, so many mutations build up within that A prime population that now it's its own bona fide species. It's now species B, and at the ancestral homeland, we still have species A. But if we were to reintroduce members of species B to species A on the homeland, they would no longer be able to interbreed. We had an allopatric speciation event. By contrast, looking at vicariance, this is when a natural situation arises, like a river opens up or we have a mountain building event. Here we have species A on our ancestral population mainland. We then have the opening up of a river that divides species A into two populations. We've got species A prime and species A double prime. Over time, because of lack of gene flow across this physical barrier, those mutations are going to build up so much that we're not going to have compatible reproductive events between these two species, and therefore we can go ahead and call them different species, species B and species C. So no longer are they considered species A, they are now considered two distinct separate species formed by this allopatric speciation event. Another recent example of allopatric speciation is with the wrasse fish. Now this is even more recent than the splitting of the subspecies of the owl in the western part of the United States. Here we have what we now have as two distinct species of wrasse that inhabit the waters of Central America around the Isthmus of Panama. The Isthmus of Panama is a recent geographic feature that was lifted out of the sea, and it acted to form a physical barrier between populations of wrasse fish. And over many thousands of years, that physical separation allowed an allopatric speciation event between the blue-headed wrasse and the Cortez rainbow wrasse. Now with allopatric speciation, we also have something called hybrid zones. So in the intermediate time between the formation of bona fide species, you can still have some contact and gene flow between those two populations in what we call the hybrid zone. The hybrid zones occur from secondary contact, and if the two groups later come into contact, hybrids can be produced. Now, as we discussed previously, some hybrids don't make it. Some hybrids are able to reproduce on their own, but aren't able to compete with those bona fide species. So in the hybrid zone, we have the formation of those hybrid species. Here we have an example of two species of oriole, the Baltimore oriole and the Bullock's oriole. They're able to form hybrids in this hybrid zone here in the light orange color, where they are bridging the two distinct species of the Bullocks and the Baltimore. Several situations can arise through the hybrid zone. So changes in the hybrid zone over time can be summed up in three ways. So one, we can have reinforcement. We can have hybrids that are less fit than either purebred species. The species are going to continue to diverge from each other until the hybrids can no longer occur. Okay, so that is reinforcement of this speciation event. Next, we can have fusion. And this is when reproductive barriers are weakened until the two species become one again. So this is the loss of the hybrid zone while those two populations were on their way to becoming new species. We had fusion in the hybrid zone, and therefore we produced the same interbreeding population. Next, we can have stability. And this is when the hybrids are fit enough to continue to be produced. So their fitness is okay, they're able to adapt to their environment just fine, and therefore we have a stable hybrid zone while these two populations are continuing to diverge, ultimately resulting in a speciation event. Now, we should talk about some terminology and understand that hybrids are not to be confused with ecotypes. And what I mean by an ecotype is a genetically distinct geographic variety or population within a species that is genotypically adapted to specific environmental conditions. So they're of the same species, but they represent a distinct population, and we call that distinct population an ecotype. And getting into a case study of an ecotype that I found this morning, we can talk about Latornina saxtilis, a case study in ecotypes of snails. So we have this researcher, Kirsten Johansson, at the University of Gothenburg in Sweden. She was very interested in the different polymorphic snails that she was observing on these Swedish islands. She identified through the course of her research at least 4 ecotypes of one species of snail that are each linked to specific shore microhabitats. So each population of this snail is distinctly adapted for specific places on the shoreline. So we have over here a boulder habitat, and this is what the S ecotype looks like. So the S ecotype is living specifically only in the boulders of the shoreline. And then we have a different ecotype, the E ecotype, which is specifically adapted to inhabit the cliffs on the shoreline of Sweden. Okay, and in between the S and the E, we have these viable hybrids that are formed. And we call this species polymorphic because it takes on many, many different shapes, sizes, and forms as depicted here in this image of the different types of shells that these snails use. And Johansson proposed in 2010 the mechanisms that facilitate such ecotypic variation. Number one, we have independent new favorable mutations in the different sites. Okay, so the S ecotype is specifically adapted to inhabit and thrive in the boulder environment. We have the E ecotype, which is specifically evolved to adapt to the cliff ecotype. Next, we have recombination and divergent selection from pre-existing genetic variation. So the S and the E ecotypes, as they remain separated, they're continuing to exhibit divergent selection and recombination within their little population. Now, what's interesting is we can ask, well, is this physical separation of different ecotypes going to eventually result in a speciation event? Did we capture this population at such a time that it's right before speciation occurs? And that may or may not be the case. We still have to monitor this with time, but there are lines of evidence that suggest that this will be the case. First, we have assortative mating within the different ecotypes. And assortative mating can be defined as mating preference within the different ecotypes. Specifically, in this type of snail, prefer specific shapes and sizes of their females. And this can be observed both in the field and in the laboratory. So there's some reinforcement of these two different populations remaining separated from each other. We also have restricted gene flow between the S and the E ecotypes. Even though we do have a hybrid zone, it is not as robust as some other hybrid zones. So this restricted gene flow may result in reproductive isolation and eventually speciation between the two ecotypes. And I just thought that was extremely fascinating as an example in a case study of ecotypic variation and how it might progress to a bona fide speciation event. Next, we can talk about archipelagos. These are simply island chains. We have the archipelago of Hawaii. We also have the archipelago of the Galapagos Islands. This all falls to a field of biology called island biogeography, which is a study of speciation on islands. We've already discussed the founder effect, but the founder effect plays a huge role in island biogeography, where we have few individuals of a species from the mainland arrive on other islands. And over time, because of that geographic separation, allopatric speciation is going to occur, and it results in high amounts of speciation as new species occupy different available niches on those islands. And if you were to reintroduce those different populations to each other again, they wouldn't be able to interbreed because so much time has passed, they've had a buildup of mutations that they can no longer interbreed. So we've been talking about speciation that occurs with a physical barrier in place, but we can also have speciation events that occur where there's no physical barrier in place. Parapatric and sympatric speciation occur within the same habitat range. Parapatric speciation and parapatric populations occupy distinct niches in their environment, and thus they are separated not by physical barriers, but by ecological factors. And we can break this word down. Para means near, and patric we know means homeland. So this is speciation that occurs near the homeland. This is often associated with those clinal gradients that we were talking about earlier, whether it has to do with altitude, humidity, different mountain slope environments, or human impacts. Sympatric species share the same ecological niche. Let's break this down again. Sym, S-Y-M, means together. So this is together on the homeland, there's a speciation event. And we've used this term ecological niche. An ecological niche is the role and position that a species has in its environment. This can do with what it uses for food, nutrition, and shelter, the strategies that it uses to survive, and its mode of reproduction. Taking a look at an example of sympatric speciation, this occurs within a population. Here we have the cichlid fish, which all evolved from one individual species. These fish occur in Lake Victoria in Africa, and they evolved within the same habitat and the same geographic area. But what drove their speciation from that one ancestral species is food specialization. So we have different specialists, some that eat snails, some that are algae scrapers, some that are leaf eaters, and therefore they all diverged on different paths, and therefore we had an event of sympatric speciation. Another example in action today is the apple maggot fly. There are two groups of apple maggot flies, one that lays eggs on hawthorns and one that lays eggs on apples. Males will look for mates on the same type of fruit that they grow on, and females are going to lay their eggs on the same type of fruit that they grew up on. Therefore, flies that grew up on hawthorns will raise offspring on hawthorns, and flies that grew up on apples are going to raise offspring on apples. So we already can see genetic differences between the two groups, and over a long period of time, they could become separate species on their own right. So this is another example of sympatric speciation that's occurring right now that we can observe. This week we have a TED Ed video by the Amoeba sisters going through different speciation mechanisms and putting a graphical representation to what we spoke about today.